Thanks, Matt. And good afternoon, everyone. Thanks for inviting me here. I'm actually en route home from a wedding in Michigan this weekend, and we've left my 2 teenage sons at home since last Friday on their own, and so you'll excuse me if I give my talk and then leave for the airport, because I'm not sure what awaits me at home, but I'm sure nothing could go wrong with 2 teenage boys at home alone for 4 days. Could there? Anything? So I'm going to talk a little bit today about this large bovine respiratory disease project that I've been involved with in a large collaborative group that's working together, and this is a project that's led out of Texas A&M, so Jim Womack there is the PD, and it involves collaboration between basically 6 different universities, so Washington State, UC Davis, University of Missouri, Colorado State, New Mexico State University, and Texas A&M. But I always like to come from California, and there's always new ideas coming out there, and so there was one I saw on the internet yesterday that I thought I just had to share with my colleagues here in Nebraska. And I hadn't really ever thought about this before, but if we each grew a large crop of different food, we could all trade with each other and eat for practically free. Had you guys ever thought about doing something like that? So food is free. So there we go. We've got all our problems solved. But I had to laugh at the little snide comment from this particular individual who said, yeah, and since some people are better at growing food than others, we could have them do it so we could do what we're good at. And we could trade for things that we make food, and we could call the people who are really good at growing food farmers. And so that's good. At least we're getting convergence there on the different attitudes. But I thought that food was free is kind of an interesting concept. So that's news from California for you guys. So the long-term goal of the BRD CAP, as it's called, the Coordinated Agricultural Project, is to try to reduce the incidence of bovine respiratory disease, or BRD as I'll call it from now on, in both beef and dairy cattle by basically capitalizing on advances in genomics. And if you think about the fact that it was only 2009 when we first developed the 50K chip, it's pretty breathtaking, and this group particularly has been involved from the bloody beginning of this thing, and it hasn't always been a pretty orchestrated evolution over time as we've come to understand these processes better. And even during the course of this grant, I think the technological developments in sequencing have actually really altered how the grant is even progressing, and it's almost hard to write a grant that keeps up with what the technology is doing, and that's certainly impacted this particular project. But we've always known that the potential benefits for genomics are greatest for the economically important traits that are difficult or expensive to measure, and feed efficiency, as will be discussed in this one, is certainly one that's like that. But I would argue also disease susceptibility is one that's like that, because this is really not an experiment you want to do with your own herd. And so is there a way where we can actually have better predictability of which animals are going to be less susceptible to disease? And can we use genomics to do that rather than having to expose our animals and then find out that in fact we had a large number of susceptible animals? And BRD is a classic in this regard. It is the most important disease in both the beef and dairy industry in terms of mortalities, and it doesn't have any selection criteria at the moment. We really have no ability to incorporate that at all into selection decisions. And the potential benefits of genomics are greatest for these traits, but the problem is we don't have that ready-made training population that we have in the breed societies, where we've got other traits that are measured routinely, weights and carcass characteristics, for example. We've been able to develop genomic prediction equations for that, but we haven't been able to for this trait because we don't have these phenotypes in hand. However, if we were able to do that and select for animals that are less susceptible or more resistant, I don't like to use the term resistant because these are not going to be bulletproof animals. They're still going to have to be managed and vaccinated, but it's reduced susceptibility is what we're really targeting for. But if there is susceptibility that's genetically linked, if we're able to select for the animals that are less susceptible, obviously that's going to have a benefit to producers and feedlotters in terms of healthier animals, but also it's going to address some of the concerns related to antibiotic use in animal agriculture. And so even if we're able to get selection criteria that explains not 100% of the genetic variation in susceptibility, but even 20% would be a very valuable trait to incorporate into selection decisions, just given the $1 billion cost associated with this disease annually. And so that's the kind of the overall hope. The problem with developing a project like this is that we need these large training populations. In the same way the different breed associations have struggled over the years to get together 1,000 animals that have genotypes and phenotypes for different traits, we're similarly going to need large populations. And where are we going to get large thousands populations of animals that are well phenotyped for bovine respiratory disease? And that's basically what this project set around to do, was to try to develop these large populations so that we would be able to tease apart the genetic variation for this relatively low heritability trait and find out what markers of the genome or what regions of the genome were associated with disease resistance or decreased susceptibility. And so we knew we were going to have 7,000 of individuals, and that's not a population that's sitting in any university's back pocket. And so as a result, we really collaborated with commercial facilities where we had access to very large numbers of animals to put together the populations that we needed for this project. One of the things that's really problematic with this particular disease is it's not trivial to say for sure whether an animal has bovine respiratory disease or does not. And so we were really very cautious about how we were going to define an animal. So typically an animal might get pulled in a feedlot based on the pen rider's ability to pick a depressed animal, or maybe an animal that's off feed. But that is a very subjective kind of a pull, and some animals might not be feeling well with respiratory disease but put on a good face so as to not draw attention to themselves and not get pulled even though they are actually sick, and vice versa. And that type of field data is very noisy because you basically bring animals into your case population that aren't sick, and you bring sick animals into your control population, and that really dampens down your genetic signal, and so it's much harder to tell which genomic regions are associated with animals that are sick versus healthy. And all of that effectively decreases the heritability because you have all this environmental noise in the background that makes it harder to tease out that genetic signal, or what proportion of that trait is associated with genes versus environmental factors. And obviously this disease is very influenced by environmental factors like vaccinations and shipping distances and all of those types of things, which is why it's called shipping fever. And so we knew all that going into it, and so we were very careful at the beginning of this project to have a very defined criteria that we used to say whether an animal was sick or not. And basically it was designed after the Sheila-McGurk scoring system that's shown on this little card here. And if you look, there's several different factors that get taken into account. So temperature in the blue lines there, cough, nasal discharge, eye scores and ear scores. And if you're a 0, you're basically normal for those things. So you have a normal temperature and normal all of those signs. And then it progressively gets worse, such that a 3 is a very sick animal with regards to those signs. And basically what we did is an animal to be a case, or an animal that's classified as sick on this experiment, had to have a score of at least 5. So a perfectly healthy animal would have a score of 0. But if you had an animal that had, a 3 and a 2, or 3 2s, then they would get over the 5, and then they would be classed as a case. And so it was a very objective approach to try to pick out sick animals. And I have some pictures here of dairy calves that you probably don't even need to do the scoring system on to have a look at them and tell quite quickly that they're animals that are suffering from respiratory disease. And so there were 2 large populations that were in this project. There was a dairy population, and that was carried out by some veterinarians in California at this large dairy calf ranch in California, which houses about 70,000 head in these little kind of chicken coop looking things, which actually are calves in there. And what that enabled us to do was walk down the rows every morning and pick an animal that was sick, and then if it was the case that the animal was sick, then we would pick the neighbour of that animal as the control. So the hope was there that we would have animals with very similar environmental exposure to whatever pathogens were associated with respiratory disease, but we had an animal that's sick, and then right next door an animal that had the same environment that was healthy. And so it was really the perfect position to do that type of a large trial, and it was carried out by a veterinarian internship. And so what we did was collected blood from these animals, did deep pharyngeal swabs where we went past the nasal into the pharynx to try to isolate which bacteria were associated with those calves. We did nasal swabs and did virus analyses on all of those. And if you want to see what a deep pharyngeal swab looks like through a lateral cut, that's basically what we're doing. So going right back in the nose there. And basically, this is a much less invasive system than having to do bronchial alveolar lavage, which is kind of the alternate approach. And so we did all of this on over 2,000 dairy calves, but also on 2,000 beef animals in feedlots. And all of that's quite a big ask for the commercial feedlots that were involved in this project. So in the case of the dairy calves, we had the same evaluation done on them, so they had to get a score of 4 or less. And as I mentioned, we took the ones in the adjacent hutch to try to have very similar environmental conditions. And the idea is to try to have a very constant environment so that we would be able to dampen that environmental noise that's associated with bovine respiratory disease diagnosis and really up the genetic signal. That was the overall design of the experiment. So everything I just said for dairy was similarly done in beef cattle. And so we had 2 collaborating feedlots that worked with us. One was a feedlot in Colorado where we got 1,000 case and 1,000 control animals. And I got to give credit to the feedlot. If you think about the fact I told you we needed pen mates that were healthy, that were in with sick animals, what that meant is they had to go back and get 1,000 pen mates, perfectly healthy pen mates, and run them through the chute and do that entire battery of collections on them. And then keep those animals together so that we got all of their slaughter data as well. And so that's a big ask for a feedlot. I'm sure you can imagine that. And so I really give a lot of credit to the feedlots that were involved. This one's the one in Colorado, and then we worked with Par 4 cattle feeders in Washington to get the same data from those animals. So all of that data has been collected and the DNA has been extracted, and then a group has run studies on that. So in California on the dairy ranch we had about 200,000 calves that were screened in order to pick our 1,000 that were sick and then get the matching 1,000 control animals with that. And fortunately and gratifyingly, we found that there was a significantly higher level of the BRD pathogens associated with the case groups. So that suggested that we were actually doing a good job at picking the sick animals. And we have all of that data. And then actually Jerry Taylor, who's sitting here in this room at Missouri, Chris Seabury at Texas A&M, and Holly Nybergs at Washington State all analysed the data using various GWAS type programs and came up with heritability estimates in the 20% range, which again is quite good for a disease trait, which suggests that our efforts to try to minimise background environmental noise worked quite well. And they're finding that about 20% of the genetic variation could be explained by the GWAS analysis that was done in the dairy animals. The beef study, we don't have yet the GWAS data done. They have done preliminary analysis on the Colorado herd and are getting similarly reasonable heritability estimates from the data that's coming out of there. And so that's very hopeful because it suggests we are pulling out regions of the genome that are associated with respiratory disease. And the belief is that, you know, that the experimental design here is helping to increase those estimates. So the future plans for the dairy study is to try to narrow in on the regions that are showing up in the GWAS as being regions associated with less susceptibility to respiratory disease, and try to use some of the more recent sequence information to try to identify causative mutations or the actual SNP that's resulting in that animal not getting sick to disease. That's really the holy grail of this experiment, and it's really something that we didn't have available at the beginning of this experiment because we just really didn't have sequence data, and I'll talk a little bit about how we're going to go about doing that. And then of course what we need to do is incorporate this information into genetic selection somehow, and so how is that going to be done? And so Kurt Van Tassel at the USDA ARS is working to try to integrate the prediction equations from these Holstein dairy calves into what's called the net merit evaluation, or the index that the dairy cattle producers use, which at the moment of course includes things like milk production and mastitis and health traits, but it doesn't include bovine respiratory disease, and so it's going to need to be incorporated into that index at the appropriate weighting for that particular trait, and so how much of selection pressure should go on that. And ultimately it needs to get into there at the right weighting for that, and that's the similar thing that will need to happen for the beef industry to adopt this as well. So with regards to the beef study, they're currently working on doing the genome-wide association study, that is having a look at which regions are associated with the susceptibility and having a look how much variation that explains. And the other part of that I mentioned a minute ago is that Jerry Taylor has worked in collaboration with GeneSeq to develop a 250,000 SNP chip. So we've talked about the 50K, that's 5 times less than this 250,000 SNP chip. But as Dr. Pollack alluded to earlier, looking for functional SNPs. In other words, SNPs that are in important regions of genes that where a base pair change is expected to cause that protein to be malformed or truncated early and not functional. So we're looking for actual functional mutations that might be associated with, for example, a protein folding a different way so it's no longer susceptible to maybe a virus attack getting into the cell or things along those lines. And so what's happened is this 250K functional chip has been developed based on the sequence data that's recently become available in the case of over 400 animals from multiple taurine breeds and that have also been involved in this 1,000-bull genome sequencing project. And the hope there is that we'll actually, rather than having a marker that's kind of indicative of this is the region associated with BRD, we'll have the actual SNP that causes the mutation in a protein that makes that animal susceptible to BRD. And that is meant to be a lot more predictive across generations and hopefully, for you guys, across breeds than is a marker that's just kind of in the general region of that piece of that genome that's associated with BRD. And so we're really looking for the holy grail functional mutants that are causing actual changes in proteins that are actually causing the phenotype that we're interested in this case, bovine respiratory disease. And that is all in place and things are scheduled to start getting genotyped in November. And then, of course, with beef cattle, we have many different breeds that we're working with and many different genetic evaluation providers, breed associations, that are working to incorporate this information into EPDs, because that's the way that this has to come into beef cattle evaluation and genetic selection. It can't be a separate piece of information. We've tried that experiment and it didn't work very well. And so I think there's kind of two thoughts as to how this might come down. So if in fact we get to the end of the experiment and it doesn't look like there's any actual causative functional mutations that are explaining a reasonable amount of the genetic variation, then we're going to have to go with the same route that's been used for predicting traits that we do to the current date, which is to get breed-specific bovine respiratory disease populations where we can actually develop prediction equations for this disease within breeds. If, however, and this is what we're hoping is the fact, there are actually some large single effect loci where we actually are able to identify a SNP that's associated with a protein that's actually causing susceptibility to disease, then it's hoped that we can actually use those SNP calls to incorporate that into genomic predictions and that those causative mutations are actually going to be useful across breeds. And we don't know the answer to that question. That's a hypothesis, if you will, at the current time, and we need to try to find out whether or not that's the case. And so, you know, if it's polygenic, in other words, there's no single SNP that's causing big changes in a protein that's affecting susceptibility, then we're going to go the usual route, if you will, of how we've been incorporating the genomic test result up until this point, which is determination of some type of an accuracy or correlation between our genetic test and the trait of interest, and we'll incorporate that into genomically enhanced EPDs in a breed-specific fashion, the same way that we do for marbling or weaning weight or whatever trait we commonly do. That, however, requires breed-specific data on BRD phenotypes, which is not something we currently have. The animals that were involved in the beef feedlot studies were just feedlot crossbred animals. They weren't specific breeds. So that is going to be the outcome if we aren't able to find these SNPs that have effects on genes that have it can explain a reasonable amount of the genetic variation. The hope is that with those SNPs we can actually just incorporate them in based on the SNP calls, and basically the way that looks is you now kind of add in the red piece of information here where the actual functional variants start to get incorporated into the genetic evaluation, and they feed into the test animal's genotype, and you get a genomic breeding value based solely on, let's just say, the 10 SNPs that are associated with functional changes in the proteins. And so we don't know what the answer is. We're still in the middle of analysing all of this data, and so I hope that by this time next year we'll be able to give you a firm feeling as to how exactly that's going to turn out and what the genetic architecture looks like. If it is that there's some SNPs that are associated with a reasonable amount of genetic variation, that would be the easiest way to incorporate things into the genomic predictions. If we need breed-specific predictions, as we have done for traits up until this point, then that's going to be a much longer story in order to get there. So that basically is what, if they're large effect causative functional mutations, then their effects should persist across breeds, and that would be the ideal situation. Otherwise, we'll need to develop prediction equations for each breed, as we have up until this point, and developing an ongoing phenotyping program. And that's problematic because the reason we're doing this project is we don't have BRD phenotypes at the current time. However, if you do go to the feedlots, they actually collect a lot of BRD phenotypic data on their pools. They collect which animals are pooled, they collect whether they're treated or not. And there's a thought towards how can we better integrate the feedlot data that's getting collected into genetic evaluations, because at the moment there's kind of a total disconnect between feedlot phenotypes and those phenotypes coming back to the breed associations. And so there's a subgroup that's been put together to work with the Beef Improvement Federation to develop a set of guidelines as to what data should be collected at the feedlot as it results to BRD, that we could start collecting that at the feedlots as animals are getting sick, such that we can start bringing that information back into breed association databases and start informing those breed association databases with phenotypes associated with BRD. And so that's going to be an important thing to accomplish, but the phenotyping effort can't be greater than the expected subsequent benefit. And unfortunately, that's kind of a little bit of a hard nut because if you're doing all the phenotyping in a feedlot and then giving all that information to the breed associations and then that's all going back to the breeder, it's kind of hard to connect all that together so that there's value associated with the phenotype collection that will actually get good phenotypes. And then the other tricky part is how do we incorporate BRD susceptibility into selection indexes, or what weighting should you give to BRD susceptibility when you're making selection decisions? And I think it's a little bit more difficult in the beef world to appropriately weight traits that may not bring direct value back to the person making the selection decision. So it's the old kind of disconnect between market signal and where the value gets derived back. But it may not have immediate value to the cow-calf producer if the main area we're getting BRD is in the feedlot. So is there a way that there can be kind of vertically integrated partnerships that reward producers for including BRD selection in their selection decisions, because presumably they're delivering a lot of cattle that are going to have less problems with respiratory disease genetically as a result of that selection. And so it's a little bit more tricky, I think, in the beef industry than it is in the dairy industry to get that incorporated at the right amount. But that's one of the things this project is looking at, is what is the appropriate economic weighting in an index for this particular trait. So I didn't say that we were targeting the low-hanging fruit of selection in beef cattle, and I think that disease is probably one of the most difficult ones to really get a handle on, but it is a very valuable trait, and if we are able to get markers that are predictive for this particular trait, I think it has tremendous value, especially in this current environment of decreasing use of any type of antibiotics in animal agriculture, maybe at all, then if there anything we can do to try to genetically improve animals to be disease resistant is going to be something that is going to be of long-term benefit to the industry. And so with that, I've just wanted to acknowledge the team that's involved in this research. As I mentioned, it is a large multi-institutional research collaboration, and all of the individuals there on the right make up the research team. There's also extension and educational components that I haven't really touched on here, just as I really just focused on what the research group has been up to. And I hope by this time next year I can give you a firmer answer as to whether or not we are finding genetic markers that are associated with a reasonable amount of genetic variation around bovine respiratory disease. And I promised Matt I would leave time for questions, and I have done that, and so I'm happy to take questions at this time.